to Lecture 10 and 11. This lecture will provide your overview of cellular respiration. Cellular respiration involves many oxidation-reduction reactions or redox reactions. When we're talking about metabolism or photosynthesis, we are going to be looking at energy transfer within the cell, and usually this energy is going to be transferred in the form of electrons. As a result, both of these reactions will involve moving electrons throughout the cell and making and breaking different compounds. In general, an oxidation reaction involves a loss of electrons, whereas a reduction involves a gain of electrons. One way to remember what is being oxidized versus what is being reduced is to think of reduction as the change in charge number. When you're gaining electrons, you are gaining negative charge, which means that your charge is going to be more negative as a result. When you are being oxidized, you are losing electrons, which means you're losing negative charge, and then you usually have a positive charge associated with that atom. Another way to help remember what is being oxidized versus what is being reduced is typically when you have a gain of oxygen, you have oxidation occurring. This is because when you gain oxygen, oxygen's going to be holding those electrons closer to itself, meaning that the respective atom that oxygen is binding to is going to be oxidized, losing those electrons. A loss of oxygen is typically associated with reduction. Conversely, a gain of hydrogen is usually considered a reduction. This is because hydrogen is usually carrying extra electrons. And when you have a loss of hydrogen, this is usually considered an oxidation because as you lose hydrogen, you're also losing electrons. Another tip to remember what is being oxidized versus what is being reduced is remembering that hydrogen is usually used as an electron carrier. This is because when hydrogen is present, we usually have extra electrons, and in that case, it forms what is termed a hydride ion. So an important concept to review is the importance of hydrogen in these pathways. We've seen hydrogen in the form of a proton. That would be H+. In that case, the hydrogen has lost electrons and become positive to lose the electrons and to become that H+ ion. However, in this case, when we are looking at metabolic pathways and photosynthetic pathways, when we have that hydrogen atom, in this case, it will gain an electron. So remember that hydrogen only has one electron in its valence orbital. So it can give it up and become a proton, or it can also take an electron in and become stable because it can only hold 2 electrons in that first shell, that first orbital. When hydrogen gains an electron, it becomes an electron carrier, and it also becomes H- or the hydride ion. So this is very important to understand. Hydrogen as an atom itself has 2 options. It can lose that electron, become a proton, or it can gain an electron and become a hydride ion. In these metabolic and photosynthetic pathways, one of the major roles of hydrogen is an electron carrier, so it would be in the form of that hydride ion. This is an example of what a redox reaction looks like. Keep in mind that when you have an atom in its elemental state, it has a charge of 0 or is considered to be neutral. So in this case, we have magnesium in its neutral state, meaning that it has a 0 charge. We are going to add oxygen to it. Once again, oxygen gas, O2. We don't see any charge associated here, so it would be considered to have a charge of 0. We have the reaction occurring and we're forming magnesium oxide. In this case here, we've got magnesium, which now has a 2+ charge associated with it, and the oxygen has a -2. So with respect to magnesium, we have gone from 0 to +2. This means that we have had a loss of electrons, or magnesium was oxidized. Oxygen went from 0 to -2. This means that oxygen, in this case, has gained electrons and thus it has been reduced. Here is another example of an oxidation reduction reaction. In this case, we have methane, CH4, being oxidized to CO2. This is a typical combustion reaction. Methane gas plus oxygen gas will lead to the production of carbon dioxide, energy, and water. So if we take a look at the carbon and what is happening to the carbon here, we have carbon with CH4. We are going to have it form eventually CO2. So let's take a look at these electrons and how it is shared in these molecules. Methane gas has CH4, and all of those electrons are being shared equally between carbon and hydrogen because these are non-polar bonds. We then have that carbon now being used to form carbon dioxide. If you take a look at the third molecule in the blue diagram, we have the formation of carbon dioxide. Notice here that because oxygen has such a high electronegativity value, those electrons in that bond are going to be held closer to oxygen and further away from carbon. In essence, that means that carbon will have become oxidized. Those electrons, even though they haven't really left carbon, they're going to be held further away from carbon. So carbon is going to feel like it has almost lost those electrons. If we take a look at oxygen, we have oxygen gas. Once again, those electrons are going to be equally pulled between those oxygen atoms, so that is considered to be nonpolar. However, we then have it forming water. So in the formation of water, we now have oxygen holding those electrons closer to it. They, in essence, become reduced. And those hydrogen atoms are going to be donating their electrons, not completely, but they're going to be giving their electrons up to oxygen because they have a very low electronegativity value. Here we have our typical glucose molecule, C6H12O6. We are going to add oxygen to it, and once again, this is a combustion reaction, so we are going to form carbon dioxide, water, and also energy. In this instance here, we have once again, the carbon and the sugar will become oxidized. It will form carbon dioxide, and those electrons will be removed from the sugar molecule. Oxygen, in this instance, will be used to form water. Once again, it will become reduced by taking in extra electrons. Overall, this reaction is considered to be negative, very negative. It releases over 686 kcals per mole. So there is going to be a lot of energy released in this reaction. This is the overall reaction for your aerobic respiration. Respiration essentially is a catabolic reaction. There are two different pathways for sugars that sugars can take within the cell, aerobic and anaerobic respiration. Anaerobic respiration is also considered to be fermentation. No oxygen is going to be required in anaerobic respiration. And in this instance, we would have the partial degradation of the glucose molecule. As a result, there is much less energy harvested in anaerobic pathways. Aerobic respiration, on the other hand, uses oxygen, and in this case, we have complete oxidation of all the carbons to carbon dioxide. This means that every single carbon in that sugar molecule is going to be oxidized. Those electrons are going to be stripped away from the carbon and to be used to eventually make ATP molecules. In aerobic pathways, much more energy is going to be harvested, so the aerobic pathways are considered to be much more efficient at harvesting the energy from sugar molecules. These catabolic reactions, when we talk about them releasing energy, we are going to be talking about them in the form of high energy electrons. So this energy is going to be transferred between these reactions from the movement of these electrons. And this is going to be done through a series of oxidation reduction reactions. Let's take a look at the energy that can be in the form of electrons. Remember, the electrons in the carbon-hydrogen bonds are considered to be nonpolar, meaning that they are shared equally between those two atoms. However, These electrons are considered to be high energy, meaning that they have a very high energy state associated with them. Why is this so? Because pulling electrons from a more electronegative atom and giving it to a less electronegative atom will require energy. This is what photosynthesis actually does. And we will see we'll understand that more when we get into the photosynthetic process. But overall, when you're passing electrons from a less electronegative atom to a more electronegative atom, so let's say we are passing it to something like oxygen, this typically will release energy, and this is what we see in our aerobic respiration pathways. Remember, in these pathways, we're going to see the hydrogen atom as the electron carrier, specifically in the form of the hydride ion. In metabolism, these electrons are going to be passed to other molecules in a series of small steps. This allows for the cell to optimally harvest the energy from this process. Electrons are going to be taken from glucose and they're going to be passed to an electron carrier, specifically NAD+. NAD+ is an important electron carrier in the cell. NAD+ is also considered to be a coenzyme and a nucleotide. As the glucose molecule is oxidized, those electrons are going to be taken from the glucose molecule in a high energy state. They're going to be delivered to NAD+. As a result, NAD+ is going to be reduced because it is taking in these electrons, and it is going to form NADH. So NADH is the holding place for these high energy electrons until we get to the later stages of metabolism where the ATP is actually going to be produced. The transfer of these electrons are carried out by a dehydrogenase enzyme. As a result, we are going to also have the release of a proton that is also going to be released from glucose. We will see how that occurs in the next slide. So NAD+ has the ability to carry 2 electrons. These 2 electrons are going to be carried by 2 hydrogen atoms. So remember, when hydrogen eventually carries these electrons, they will form the hydride ion. But in this diagram here, we have them separated out as 2 electrons and 2 protons, simply so that you can see where the electrons and where the hydrogens are actually going in this reaction. These electrons and one of those hydrogen atoms are going to be transferred to NAD+ to form NADH. We can see in this diagram here that we've got glucose molecule, we've got extra electrons that are going to be taken from the food particles. We have the dehydrogenase enzyme that will come in and help to catalyze this reaction. So these 2 extra electrons are going to be added to NAD+, as well as one of the hydrogen atoms. The other hydrogen atom will be giving up those electrons. It will form the proton, and that will then be released. So here we've got the reduced form of NADH. Once again, it carries 2 high energy electrons in this process. Also notice that the dehydrogenase enzyme will catalyze the forward and the backward reaction. So NAD+ can be used to form NADH, and NADH will eventually lose electrons and become oxidized and can return to the NAD+ form. This diagram here shows you the importance of carrying out these reactions in a series of steps. If we were to have the overall reaction of hydrogen bonded to oxygen to form water, this would release a tremendous amount of energy, and this would be very difficult for the cell to harvest. However, through the use of the electron transport chain, we can transfer and move these electrons through the transport chain, harvesting the energy at each step to eventually maximize our production of ATP molecules. We have the same overall reaction of hydrogen being bonded to oxygen to form water. However, we have optimized the ability for the cell to extract and make ATP molecules. There are 3 different phases of respiration. The first phase is glycolysis, where we have the initial breakdown of one glucose molecule into two 3-carbon pyruvate molecules. This occurs in the cytoplasm and does not require oxygen, so it is considered to be an anaerobic step, even though it is part of the aerobic pathway. We then have the citric acid cycle, which is also known as the Krebs cycle or TCA cycle. This is where we have the complete oxidation of pyruvate. So every single carbon in that pyruvate molecule is going to be stripped of its high energy electrons. This occurs in the matrix of the mitochondria. We then have oxidative phosphorylation. This is where the energy from those electrons is going to be used to generate ATP molecules. This is done in a two-step process. The first step is where the electron transport chain will be used. The energy from these high-energy electrons will be used to pump protons into the intermembrane space. The second step of oxidative phosphorylation is where the ATP is going to be made. This is where we have ATP synthase come in that will take advantage of the proton gradient to then create ATP molecules. This also occurs in the mitochondria and specifically the inner mitochondrial membrane, and it also involves the matrix and intermembrane space. This slide gives an overview of those processes. Glucose can come in different forms, either through photosynthesis, ingestion, or absorption. Glucose will then undergo the process of glycolysis, where it will be broken down into the pyruvate molecules. These pyruvate molecules will then enter into the citric acid cycle. Notice that in glycolysis, we do have the production of ATP molecules. However, there is not a lot of ATP produced by this step. We will have the citric acid cycle, where the main goal is to make those electron carriers such as NADH and FADH2. The citric acid cycle will also produce carbon dioxide as a byproduct. Then the electrons that are going to be carried on NADH and FADH2 will then undergo the oxidative phosphorylation phase. This is where the bulk of ATP is going to be made. This will end with the utilization of oxygen, which will eventually be used to take in those lower energy electrons to then form water at the end of this pathway. We will begin with a breakdown of the glycolysis process. Glycolysis begins with one glucose molecule. Remember, a glucose molecule has 6 carbons in its structure. This glucose molecule is going to get phosphorylated on both ends. This means that 2 inorganic phosphate molecules are going to be added to glucose. This will use 2 ATP molecules. That is where the inorganic phosphates actually come from. Once the glucose molecule is phosphorylated at both ends, it's going to get cleaved in half. This will lead to the production of 2 3-carbon molecules. Eventually, these halves are going to be converted into pyruvate. Pyruvate has 3 carbons associated with its structure. In the process of glycolysis, we are going to generate one NADH molecule, and this is going to be generated for each half of the glucose. So because we have 2 pyruvates produced, we actually produce 2 NADH in total. One water molecule is also going to be generated, so in essence we will generate 2 water molecules as each of these 3-carbon molecules get converted to pyruvate. And we will also be generating 2 ATP molecules at the end of this process. So overall we are generating 4, but remember, we needed 2 ATP molecules to phosphorylate glucose. So as a result, we have a net production of 2 ATP molecules for glycolysis. Remember, we are not utilizing oxygen in this step, we're not creating any carbon dioxide, and this occurs in the cytoplasm or cytosol. Glycolysis can be broken down into 2 different phases: the energy investment phase, where ATP is used to tag the molecule to be broken down into 3-carbon structures, and the energy payoff phase, where that 3-carbon structure is going to be isomerized to form a pyruvate molecule. And in this phase, we're going to produce ATP. So let's take a look at the energy investment phase. I don't expect you to know all of the structures and all of the individual steps involved in this process. I expect you to know the overall goal and what is occurring, and I will note the important structures to know. So it is important to know that glucose is a 6-carbon molecule. In order to get it to be broken down into 2 3-carbon molecules, we have to tag it with phosphate. And this is what is happening in the first phase. So we can see in the diagram we have 2 ATP molecules that are going to be used. They're going to be used to tag the molecule. So as a result, ATP is going to be broken down into ADP, and those inorganic phosphates released in that reaction are going to be added to the glucose molecule. Note that we go from a 6-carbon sugar to a 5-carbon sugar, specifically fructose. When we have fructose 1,6-bisphosphate, we can see that we have a nice structure that has the oxygen in the middle, has the phosphate groups at the end. This is the structure that can easily be broken down into 2 3-carbon molecules. Once we have that fructose cleaved into 2, we have the production of an important molecule that you need to know, G3P, or glyceraldehyde 3-phosphate. This is very important because we will see when we get to photosynthesis, this is the sugar precursor molecule that actually links the 2 pathways together. So G3P is an important molecule to know. So glyceraldehyde 3-phosphate has 3 carbons in the structure, but it also has a phosphate group attached to it. So G3P is the beginning structure of the second phase, the energy payoff phase. In this phase here, we are going to remove those phosphates and we're going to generate electron carriers as well as ATP molecules in this process. So we can see as G3P is converted to 1,3-bisphosphoglycerate, we have the production of NADH. So NAD+, 2 molecules of NAD+ are coming in, because remember, we have 2 G3P molecules. So this energy payoff phase is occurring twice, once for each G3P. So G3P is going to donate its high energy electrons. NAD+ is going to be reduced to form NADH. We're also going to have the release of protons, because remember, only one hydrogen atom is going to be added to NADH, the other is going to be released as a proton. We will then have, once again, a series of isomerization reactions where the carbons and the base structure of that molecule are going to be shifted around. Once again, I don't want you to know the enzymes and the specific structures involved. But you should know that as this is occurring, we can produce ATP molecules as we are removing those phosphate groups. And in the step 8 to 9, we have the production of water molecules. So at the end of this energy payoff phase, we have generated 2 electron carriers, NADH. We've also generated 4 ATP molecules, and we've also generated 2 water molecules. Because remember, there's 2 G3P molecules going through this process. In the end, we have now generated 2 pyruvate molecules. Note that we have 3 carbons in the pyruvate. So overall, we've gone from a 6-carbon glucose molecule to 2 3-carbon molecules. This means that we have not lost any carbons in this process. We've simply cut that molecule in half and rearranged it. So because we haven't lost any carbon in this process, we do not have any oxygen being used, and we are not producing any carbon dioxide.